All right, so today I'm going to be talking about the genetics of FTD. I'm going to start with a little genetics lesson, followed by a history lesson, and then talk about when ALS joined into the spectrum, and then end with some discussion of genetic counseling and genetic testing. It's an echo. So here we have our lesson, DNA. It's that long, twisty, rope laddery thingy. That's all you need to know. So DNA contains the instructions for cells to do what they do. And DNA is packaged into structures called chromosomes, and chromosomes are present in the nucleus of the cell. The humans have 23 pairs of chromosomes, and you inherit one set from each parent. And your genome refers to the entire complement of DNA that's present in your chromosomes. So DNA is organized into units called genes, and each gene is coded by the letters A, C, G, and T. These are the nucleotides or bases that make up the sequence. And we all have the same 20 to 25,000 set of genes, but what makes us different, each unique, except for twins, is that there are slight variations in these proteins called polymorphisms or SNPs. So this results in proteins that are the same basically, but have slight variations. So genes contain the instructions to make proteins. The DNA is converted into something called RNA, which is the messenger from the DNA to make the protein. And the protein is made up of units called amino acids. So if you have a change or a mutation in the DNA sequence, that may change the structure or function of the protein that's produced, as shown in the box on the right. Do I have a pointer?
The top is the pointer.
Okay.
If you push.
Oh, the top.
The start.
Oh, got it. Okay. All right. So as you see here, there is a mutation, and this changes the sequence, and that results in an abnormal protein. The abnormal proteins can be changed by single letter changes or a change in the reading of the sentence. For example, the cat saw the dog is changed to the bat saw the dog, or the cat saw the hog, or the cat sat the dog. So you can see that it's basically the same, but has a slightly different meaning. In addition, some other kinds of changes result in garbled proteins. For example, if you have a deletion of a letter or an insertion of a letter. And these can result in changes in function or changes in the level of protein. In neurodegenerative diseases, many times they result from abnormal aggregates of misfolded proteins. And mutations can change the protein in such a way that it either is present at a lower level or higher level, or is abnormal in some way, and therefore results in aggregates. And these aggregates are shown here schematically, and these may become present in the cell body or nucleus and form inclusion bodies, and these result in cell death.
Anything involving dyslexia?
No. I'm not sure
You read cat, dog, sumac, all that. Sometimes you see the letters backwards.
Right, but the DNA reading machinery pretty much just goes from left to right. So it just sees a slightly different sequence. So neurodegenerative diseases are often characterized by particular proteins that are aggregated. For example, in Alzheimer's disease, you have amyloid plaques and tau tangles. In Parkinson's disease, you have deposits of something called alpha-synuclein. And those result from mutations in those genes. So Understanding genetics is important because it can give us more information about the causes of disease, for example, the mutations that cause those diseases, and it enable us to find new targets for treatment and allow us to perform correlations with biomarkers, imaging, pathology, and clinical features so that we can better understand the diseases. There's also some clinical benefits to genetic information. For example, if you know that a particular mutation is present, that can help confirm a diagnosis, and that better diagnosis can lead to one feeling more confident that you have the right diagnosis and being able to institute the right therapy and stop doing additional testing to find the diagnosis because you have it. In addition, genetic information provides information that can be used for counseling family members. And also, if you have information that can predict who will have disease, that allows for pre-symptomatic testing, and also not only from the information being given to that individual, but also from the perspective of research and clinical trials. The whole goal is to, if you can of developing all these therapies, is that you want to be able to do something, and the ideal time to do something is before somebody has symptoms. So being able to predict who is eligible for a particular clinical trial is important. So genetics is important for FTD in particular because about 40% of FTD cases have a family history of a similar disease. In this pie chart, the blue and red sections represent the familial forms of FTD. And the blue, they're divided into blue and red because the blue have slightly stronger forms of family history, so there's clearer evidence that there is a family history. And in these cases, the inheritance is something called autosomal dominant. And because this is the primary genetic inheritance mechanism for FTD, I'm going to go through this. So in order for an autosomal dominant condition, having a single copy of the mutation on one chromosome is enough to cause disease. So in this example here, the father has 2 chromosomes, and one of them has a red spot, and that one is the abnormal gene. The children of this couple will be able to have any combination of those 2 chromosomes. So there's 4 different combinations of the 2 chromosomes. And as you can see, 2 of the 4 have the red copy. So we translate this into saying that offspring of an affected individual have to have a 50% chance of themselves having inherited the disease gene. So the first gene to have been identified for FTD is called MAPTau, or microtubule-associated protein tau. And the story starts in 1994 when a region of chromosome 17 was linked to FTD with Parkinsonism. In 1997, the term FTDP17 was coined because MAPTau, or tau, is on chromosome 17, and therefore tau was a candidate gene because when pathology was looked at from these cases of FTD, they found pathology, those aggregates that were composed of tau. Subsequently, in 1998, finally, after a long search, mutations in the tau gene were identified. And tau mutations are inherited in an autosomal dominant manner. And since that time, more than 40 different mutations in tau have been identified. So this was a seminal finding because it showed that having mutations in tau was sufficient to cause neurodegeneration. But that didn't explain all cases of familial FTD. As you saw, there were about 40 to 50% that are familial. So the hunt continued for additional genes. And in 2001, a region on chromosome 9 was linked. And when I say linked, what I'm referring to is that when they look at the genome of multiple individuals from the same family, they look at markers. And what they're looking for is particular markers that are present in all the individuals who have disease and are absent in the individuals who don't have disease. So they can narrow it down to a particular chromosome, because if they look at enough people, they can see who got each part of the chromosome by looking at those markers, and they can then pinpoint it to a particular chromosome and to a specific region on that chromosome. So for a long time, chromosome 9 was linked to cases that had FTD and families mixed with FTD and ALS. And so we're going to leave that there and come back to that later in the talk, because this is a timeline. And it wasn't until a few months ago that this gene was finally identified. But a couple years later, there was an exciting finding, because it happened to be on chromosome 9, but it wasn't the chromosome 9 gene, and it was something called velocin-containing protein, or VCP. And it was for FTD, but it was in particular in a subset of FTD cases that occurred with inclusion body myopathy associated with Paget's disease of bone. And more recently, a few cases of VCP mutations have been found in families that have a mixture of IBM and ALS. So moving forward, another discovery that raised some interest was something called chromatin modifying protein 2B, or CHAMP2B. However, since then, it hasn't really panned out to be all that important, because only a few families have been found, and that was a locus that was on chromosome 3. So between tau, VCP, and CHAMP2B, tau is about 5 to 10%, but VCP and CHAMP2B, because they're a very specific phenotype, didn't really add very much. There was still an interest, a need to find additional loci. And in particular, researchers noted that there were families that had this linkage to chromosome 17, but as hard as they looked, they couldn't find any mutations in tau. And when they looked at the pathology from these cases, they found a specific type of pathology called FTLD-U, U standing for ubiquitin, and that basically meant that it was some sort of garbage protein that was being deposited, and it was tagged with ubiquitin, but they didn't know what the protein was. And unlike the other cases with tau mutations, these did not have tau pathology. So in 2006, using genetic strategies, those family linkages and DNA sequencing, a gene called progranulin was identified. And interestingly, as they suspected, because they thought it was near tau, but they couldn't find the tau mutations, and it turns out progranulin is right next to tau on chromosome 17, so somewhat of a coincidence. And again, progranulin, like tau, is found in about 5 to 10% of FTD cases, so we're up to about 10 to 20% now. Progranulin mutations are also inherited in an autosomal dominant manner, and in contrast to tau, where you get abnormal function in the protein due to a change that changes the amino acid sequence, most progranulin mutations resulted in loss of the protein through those garbled protein mechanisms, frame shifts, and introductions of new stop codons. So you would get truncated proteins what this is referred to as a haploinsufficiency, is that it's half the amount of protein, and that causes disease. And there's a lot of people studying this to try to understand how that causes disease. So coming back to our characteristic inclusions, Alzheimer's disease mutations in something called an amyloid precursor protein cause Alzheimer's disease, and you get amyloid deposits. Parkinson's disease, although rare cases, is caused by mutations in alpha-synuclein, and you get synuclein deposits called Lewy bodies. FTLD with tau has tau deposits, and those are associated with tau mutations. So what is the gene responsible for FTLD with ubiquitin? Now remember that this is the FTLD-U that we were just talking about. And that it makes sense that it should be progranulin, because, if you have FTLD with tau, you should have tau mutations. But do you have progranulin pathology in FTLD-U with progranulin mutations? The answer is no. unlike that relationship, FTLDU isn't progranulin, it's something called TDP43. And TDP43 was also discovered in 2006, and it happened to be discovered here at Penn by Virginia Lee and John Trojanowski's lab in the Center for Neurodegenerative Disease Research. And unlike progranulin that was identified through genetic families and doing sequencing and localizing it by linkage analysis, TDP43 was identified purely through biochemical mechanisms. They isolated those inclusion bodies and broke them up into pieces and figured out what the sequence was and went backwards to find what the gene was. So TDP43 is encoded by a gene called TAR-DDP, which is a mouthful, so I'm just going to call it TAR or TDP43. And TAR was initially a candidate gene for FTD because it's associated with tau, with cases that have TDP43 pathology. But actually, primarily, mutations in TDP43 were found in ALS rather than FTD. There's a few cases that have in FTD, but primarily it's found in ALS. So this was kind of intriguing, and in addition, when TDP43 was identified, it was discovered that TDP43 pathology was not only present in FTLD cases, but also in ALS, demonstrating that FTD and ALS represent a clinical and pathologic continuum. And there are some clinical findings that also support this. ALS patients, as you've heard from the clinical talks this morning, ALS patients frequently have FTD symptoms. Motor neuron disease is observed in about 15% of FTD patients. And in families that have familial disease, you often find FTD, ALS in the same family. And finally, relatives of ALS patients have about a twofold increased risk of dementia. And so what was missing here, we had a protein link, but what we didn't have was a genetic link that linked FTD and ALS, and that locus was on chromosome 9. So the hunt for the chromosome 9 gene went on, as you saw, for 10 years. And this slide, while complicated, shows a region of chromosome 9 here, and each of these bars here represents a different group's study or family study that was trying to pinpoint and narrow the region where this gene mutation was. And in addition, there were a lot of genome-wide studies, among them one that we performed here at Penn that identified SNPs, or variations, single nucleotide variations in the genome that seemed to be overrepresented in patients that had FTD and ALS over the general population. And so ultimately, in the fall, the gene was identified as something called C9orf72. It had been staring at us all along. So why was it so elusive? Why did it take so long to find? We've been sequencing it back and forth. Well, it turns out it's because it's a different mechanism. It isn't just a simple sequence change. It actually is a repeat expansion, similar to what you may have heard of Huntington's disease or Fragile X. Those are trinucleotide repeat expansions. In this case, these were hexanucleotides, 6 nucleotides repeated. Normally you have 2 to 23, and in individuals that had the mutation or expansion, they were up to like 1,600 repeats. And why was this hard to find? Well, because the basic tool that we use in genetics is something called PCR, polymerase chain reaction amplification. You have a primer on one side of the region and one on the other, and you amplify across it, and then you study the part in between, that your product that you've formed. But the polymerase doesn't amplify across really long regions. So what was happening is, if you would sequence it, you would amplify your normal allele, but the one that was expanded would just not amplify. So it would look like a person just had, 2 copies of a normal gene. So, and that actually is one of the strategies that was used to identify it, because they happened to look at all the people in families that had FTD and ALS, and they noticed that the homozygosity rate, the probability of having just a single allele was higher in individuals who seemed to have disease in these families than others. And so this led to the discovery of the C9orf72 hexanucleotide repeat expansion. And why are we so excited about this? Well, not only because it solves a mystery that's been hounding FTD researchers for years, but also because it's present at a pretty high frequency. So in a quick study that we did among our population here at Penn, when we looked at all our cases with ALS and FTD ALS, in cases that we could classify as familial, mutations were present in 40 to 50%. If we looked overall, it was about 12%. And in ALS cases that were didn't appear to have any family history, we still identified it as present in about 4% of cases. In FTD without motor neuron disease, we found it in 17% of familial cases, 21% if you look at just the subset that have FTLD, TDP for pathology, because I didn't mention, but this is also a subset of cases that, like progranulin, has TDP43 pathology, although there are ways to subclassify FTLD, TDP into different subtypes, A, B, C, and these two mutations result in slightly different subtypes, so they're not exactly the same pathology. And we also found it in 7% of overall cases. So while overall it's actually about the same as tau and progranulin, which are about 5 to 10% in overall cases, it's present in a large percent of the familial cases. And the range of these frequencies in published studies over the last couple months have quite a wide range, from the teens to the double digits, 60, 70, 80%, depending on the population being studied. And as you can imagine, this is an area of active investigation to try to understand many things about C9orf72 and how it causes disease. So just to summarize the pathology aspects, if you look at FTD, it basically results in two pathologies, FTLD TDP and FTLD tau. And the tau is associated with tau mutations and FTLD TDP with progranulin mutations. And then we have the overlap with ALS, the TDP43 subset, the sporadic, the 90% that are sporadic, and these are associated with TDP43 mutations and FUS, which I didn't really talk about because I'm focusing on FTD rather than ALS here, and C9orf72 are the mutations that really link the two groups together. So we're back to the same question we were before. Why is understanding the genetics of disease important? Well, as I've been going through, all of these discoveries have led to further understanding of FTD, to, as you're about to hear, new drug targets, and a better understanding of prognosis and phenotypes. But what I'd like to focus on now are some of the clinical aspects of genetic testing, genetic counseling, and pre-symptomatic testing. So genetic counseling serves many important functions and should always be performed in conjunction with genetic testing. One thing it does is increase the family's understanding of a genetic condition and available genetic testing options. The genetic counselor shown here, Beth Wood, is with us here today, and she's going to be with me at the breakout session. She sees most of the patients with FTD, and here she's shown using some educational tools that we've developed to help individuals and families better understand the disease. So through the counseling session, we collect and interpret family history information, explain inheritance and recurrence risk for family members, although sometimes we don't have all the answers, but at least we can lay it out as to what we know and what we don't know. And in addition, genetic counseling provides informational and psychological support. So I'm going to go through a couple questions. What is genetic testing? What is the difference between clinical and research testing? Where is genetic testing available? Who can get genetic testing? Can a healthy at-risk individual get genetic testing? If I have a mutation, will I get the disease? So genetic testing is quite broad, and there are many definitions, but basically it's testing DNA or RNA, which is what DNA gets converted to before it becomes a protein, proteins, chromosomes, to detect or evaluate genetic disease-related conditions. And there are many different methods that can be used, including DNA sequencing, which is one of our biggest tools, in part because the mutations are scattered throughout some of these genes. Genetic testing can be used for a variety of purposes. Diagnostic testing is when you test an individual who is affected to look for a particular variation or mutation that is associated with that disease, and that helps confirm the diagnosis and prevent you from keeping to look on for other diagnoses. Carrier testing isn't really relevant to neurodegenerative, to FTD because it's autosomal dominant, and carrier testing is generally used to identify a mutation that is present on one copy in a recessive disease so that you need two copies, and that's commonly done, for example, cystic fibrosis for individuals who are receiving prenatal counseling. Predictive or presymptomatic testing is testing that's done on an unaffected individual who is at risk of disease based on their family history, and in order to identify information that can help them plan their lives. And prenatal testing is also not relevant to adult onset diseases, but that's essentially diagnostic testing on a fetal sample. So when we talk about genetic testing, and because I'm involved in a lot of research, it's important to distinguish the difference between clinical and research testing. We do research genetic testing because we want to discover something. We want to learn more about the genetic conditions. We don't charge anything because we have, hopefully, grants to pay for the work that we do, or donations. There's no guarantee of results because this is done in a research lab for purposes other than providing information to patients, although in some cases we return some information to patients if they've signed a consent form. Patient identifiers aren't linked to the samples, and this is really not a substitute for a clinical test. In contrast, clinical testing is performed as part of the medical workup. It's ordered by a physician. There's a fee that's charged by the laboratory. It's not clear whether insurance will pay for it, but the laboratory issues a report. Sort of, if you send the sample, you will get a report in a timely manner, and on the other hand, it will become part of the medical record. So genetic testing is available in a variety of places. So research genetic testing is available in our Center for Neurodegenerative Disease Research, and we work on FTD, ALS, PD, and AD genes, as well as related conditions. Clinical, and it's of course also available at many institutions around the country. We're not the only ones, but I got to put a plug in for our program. Clinical genetic testing is available at what are called CLIA, Clinical Lab Improvement Amendment certified labs, and these are generally available at academic institutions as well as commercial labs such as Athena. I direct a clinical diagnostic lab in addition to the research we do, and so my lab is called the Molecular Pathology Lab, and it's at the Hospital of the University of Pennsylvania, and my lab offers clinical testing for tau and progranulin, and we're in the process of developing testing for C9orf72. So which gene should be tested and by what method is sort of a case-by-case determination, and that's an important part of the discussion that goes on with the genetic counselor. So who can get a genetic test? Well, any adult can participate in genetic research, and in fact, it's very important for us to get involvement by normal controls and family members, because this helps us understand. We're not all about just doing testing of things we know about. We're about discovering new things. And in order to understand those new discoveries, you have to be able to compare them to something and see if it's significant. So testing family members and normal controls is very important. And so we're very appreciative of people that altruistically contribute to our genetic research program. In terms of clinical testing, any affected individual can get clinical genetic testing But the chance of actually finding a mutation to some extent depends on the strength of the family history, as well as how many individuals are affected in the family, the ages of onset. Earlier ages of onset are more likely to be associated with a genetic cause, as well as the different clinical presentations of the family members, and whether or not any family members have had autopsy or testing elsewhere. That all goes into play in terms of deciding what testing to do and whether it's worth it to do the testing, because there generally is a fee, and so if there's a low likelihood of finding something, you may not want to go to that expense. And of course, testing should be done in conjunction with genetic counseling. The chance of finding something depends on family history, and these two pie charts, the one on the left shows a subset of cases that we classified as familial, and then we looked to see whether or not we found tau, C9orf, for progranulin mutations. And as you can see, more than about 55% of cases had mutations in one of those genes, but about 45% didn't. But it's still about a 50-50 chance of finding something that we know about, and in the future, with more discoveries, we'll hope to get that fully filled. However, if in families that are sporadic, and I put sporadic in quotes because sometimes sporadic just means that there's isolated cases, and there can be isolated cases for a variety of reasons: adoption, small family, early death of parents. But for the most part, if you just take all comers in that group, the chance of finding a mutation is pretty low. So how about predictive testing? As I said before, it's done on unaffected adults to determine their risk of adult onset genetic disease. But in order to do this, you can't just say, oh, I want sequencing of all these genes to find out if I'm at risk. There has to have been a previous family member with a specific mutation identified, and you test for that gene mutation only. And in the absence of any available medical therapy, it still can be, so that it is in the future, hopefully, as we get new therapies, this will become more important, because if you can predict who will have disease and you have a therapy to stop it, that's our ultimate goal. But in the absence of that, some people elect to do predictive testing because they want to make life decisions about themselves and their loved ones. It's very important, because of the potential impact of predictive testing, that genetic counseling and psychological evaluation be done before testing and after testing, before receiving the result and after receiving the result. Some important things to consider in terms of doing predictive testing are some variables which are called penetrance and variability in clinical presentations, because if you get this mutation and you want to be able to say, okay, now I know I'm going to get this disease, there has to be a link between have mutation, will get disease. But it's not so perfect. There are some issues associated with that. And that is, first of all, there are environmental impacts on all disease. So there is not only genetic factors, not only mutation, but additional genetic factors that can influence how a person responds to a particular mutation. And FTLD, that's particularly obvious because there are variable presentations even in the same family. Why? What's different about the different individuals? Why are the symptoms different? And there's a wide range of age of onset, so clearly there are environmental and or genetic impacts. In addition, something called penetrance is the likelihood that a disease will present itself if you have the mutation. And as you can imagine, in an adult onset disease, the older you get, the higher the probability, because you're now older. And for progranulin mutations in particular, as shown in the graph here, which shows the age of an individual on the x-axis and the chance of having disease on the y-axis, that at about, by age 60, progranulin mutations are 50% penetrant, and they are 90% penetrant by age 70. But that could mean that you don't have disease until after age 70, so that's an important thing to consider in terms of how you're going to feel about doing some testing at age 20 or 30. in terms of your future, that it's not always easy to predict what it's going to mean. And that's why it's important to take this into account in terms of counseling as well as evaluation of family history. And that can actually impact some of the cases that seem sporadic if one of the cases has decreased penetrance for some reason. So in summary, of familial FTD cases, we have tau, progranulin, and C9orf72 that are the big players, and they encompass about 55% of identified familial mutations and about 25 to 30% of all cases of FTD. But that still leaves more genes to be identified. And so there's a lot of genetic research that's ongoing at Penn, as well as all over the world. We have collaborators all over the world to find these genes, as well as additional risk factors, doing things called genome-wide association studies and protective genetic factors. Something that's completely revolutionizing how we obtain information and the amount of information is whole genome sequencing. So it's now possible to sequence an individual's entire genome. It's getting less and less expensive. They talk about the $1,000 genome. We're not quite there yet, but I am sure it will be soon, and that's going to change our ability to look for mutations in many genes at the same time and make it more cost-effective. Clinical genetic testing is available for most of the known disease-associated genes. And just a reiteration, genetic counseling is important to be able to understand and navigate the information and the tests. And predictive testing of at-risk individuals is available if a hereditary mutation has been identified in a family member. So I want to thank Murray for inviting me to speak, and in particular, Beth Wood, our genetic counselor, who you will be seeing at the breakout session if you come to the genetics breakout session. And that's all.